Okay. So, hi everyone. Welcome to this week SAC Club. I'm Hazel, and today with me we have Julie again. So, we both biologists here at Pediatric, and today for this club, we will together explore IBD diseases, so inflammatory bowel diseases. And for the specific topic of today, we will explore we have a comparative transcriptomic analysis of pediatric Crohn's disease and ulcerative colitis. Okay. And before we dive into today's section, I would like to say, first of all, Happy New Year to everybody.
Happy New
So
Year, everyone.
yeah, we're now in 2025, and so this club we have started this club back in 2024, and it's almost I think around like 8 months for now. And let me change the slide here. And yes, we got this club every single Friday at 9 a.m. PST, or you can see it in different time zone right here. And we also, because we have started this club for almost like half of a year now, we are now in episode 21. And you can rewatch all of our previous episodes on our YouTube page. We actually have like a playlist for that. So my college will post the link to our YouTube page right here. And then please enjoy if you want to rewatch any of the episode. Okay. Okay, so now we go back to this club. We go back to IBD, and before we go into the detail, this is the agenda of this week. So for this week, we will start with like a general overview of inflammatory bowel diseases to make sure we all on the same page. And then we'll explain the approach that we use, focusing on how we combine work and sick data for analysis. And then after that, we dive into the biological findings, looking at differentially expressed genes and enriched pathways. We will conclude by examining the cell types that are likely playing a role in development and progression of IBD. Okay. Okay, first, Julie will give us like a quick overview of inflammatory bowel diseases.
Yes. So inflammatory bowel disease is an umbrella term that encompasses a lot of different diseases, such as ulcerative colitis and Crohn's disease. There's a lot of similarities between them, but they mainly differ in their location of inflammation, for example. So in ulcerative colitis, this is limited to the large intestine or the colon. In Crohn's disease, this can occur anywhere within the GI tract. The pattern of inflammation for ulcerative colitis occurs when inflammation is continuous, whereas in Crohn's disease, the inflammation occurs in patches. They also differ in the appearance of the inflammation and the location of pain as well. In ulcerative colitis, the ulcers penetrate just the inner lining of the abdomen, whereas in Crohn's disease, the ulcers penetrate the entire thickness or several layers of the GI tract. And then bleeding as well only occurs in ulcerative colitis.
Yeah. Okay. Um, that's like a quick overview of inflammatory bowel disease. And UC and CD as the two main diseases in this like umbrella topic. And now to analyze the data, today we actually use bulk RNA-seq data. And if you used to be in this club before, you might know that we already explore IBD like several times before. But this is actually, I think, the very first time that we use bulk RNA-seq for the analysis, and not only one single data set, but we kind of like combine two data sets together to analyze together. And in here, as you can see on the screen now, we use two publicly available data sets generated by the same research group. So yes, these two data sets came from the same research group. Just to minimize the batch effects and other technical variations between data, And then one data set published in Nature Communications focused on ulcerative colitis, um, mucosal transcriptomes, investigating the mechanisms related to disease severity and treatment response. And then the other study published in mucosal immunology related to, examine the age of diagnosis, dependent immune responses in pediatric Crohn's disease. And then by combining these two data sets in our SmartBook platform, we were able to increase our sample size significantly. Actually, we got around 500 samples for this combined data set, and this is crucial for improving the statistical power of our analysis and identifying more robust and reliable differences in gene expression between UC and CD. So to give you guys like a quick look of how we integrate these two data set, I will go to SmartBook right now. So this is our homepage from our ecosystem. You can see the SmartBook, the book and analytic platform right here. We explore it like that. Okay. And in SmartBook, you can see on the left panel right here, there is a section for public database where you can search for different publicly available book analytic dataset. And today I want to focus on Crohn's disease and ulcerative colitis. So therefore I go to filter here. Under diseases, I search for the Crohn's disease. And then I also search for the ulcerative colitis in here. Okay, so after applying filters, we got like 7 total studies related to these two diseases, but today we will use 2 of them. So the first one is this one, the mucosal transcriptome uh, of the treatment-naive pediatric ulcerative colitis patients, and then the second one is the Crohn's disease. So as you can see here, if you want to reproduce our result today, you can search for similar term and then you can find these 2 dataset right on SmartBook. And after that, I will create a custom dataset based on these 2. Okay, as you can see that in the first dataset, we got like over 200 samples, and in the second one, we got 300 samples, making it a total of 500 samples. Okay, so we continue and then we create a custom dataset. Okay, and after create a custom dataset, you can create like a actual working project based on this custom dataset just to save the time. So this is the one. Okay, so as you can see, I already create like a project based on my custom dataset of pediatric IBD. So Lee A. Denson is the PI of this research group. If you want to look at their other research result. So now let me explore the dataset in here. Okay, so that is how we create a combining dataset on SmartBook. Okay, and then after combining the dataset, we perform a composition analysis to understand the distribution of samples across different categories on SmartBook. Okay, so you can reproduce this chart right here on SmartBook under the quality control. You can go to composition and then create a similar chart. So as you can see here, we got 2 main studies. And then within these studies, we categorize samples by disease status, either UC or Crohn's disease. And in here, what more important is that we actually have normal sample in here. So we can compare diseases versus sample versus normal. And then we also got the anatomical location of mucosa of rectum and ileum. And this analysis shows the number of samples in each category and their percentage of the total data set, as you can see under here. For example, we got 206 samples representing like 91% of the first data sets that are diagnosed with UC in the mucosa of rectum. Okay, and that is like the overview. We performed an overview analysis of the combined data set. Now let's dive into biological finding. So first thing first, we want to compare normal UC and CD together using ANOVA analysis to show you guys some of the commonly regulated genes in IBD. Yeah. Okay.
Okay, so here's where I can explain a little bit. So ANOVA analysis or analysis of variance, this is a statistical test that allows us to compare the means of multiple groups, not just two groups. So in this case, we compared the gene expression levels across Crohn's disease, normal, and ulcerative colitis to pinpoint which genes have a significant or statistically significant expression change across all these IBD samples. So this first gene that we're looking at here, ABCB1. It's also known as MDR. So this MDR1 expression can affect the response to certain medications and might also play a role in regulating inflammation. So we see a depressed expression of this MDR1 gene in our IBD groups. Down below here, we see IL-1R1. We see an elevated expression in our IBD groups. This is a cytokine with both pro and anti-inflammatory roles. And so this could be involved in immune cell regulation and inflammation in IBD. Oh, Hazel, I think you might be muted.
Yeah. Oh, I'm sorry. Okay, so
That's all right.
I'm changing to the next slide for you to continue on explaining the commonly regulated genes.
Yeah.
Yeah.
Okay, so here we're examining two genes from the REG or the reg family, REG3A and REG1A. So the reg family is involved in mucosal healing and regeneration, but could also potentially contribute to inflammation under certain conditions. We see in both cases or in both genes, we see an elevated expression in both Crohn's disease, but maybe not necessarily in ulcerative colitis, especially Reg 1A. The comparison between normal and ulcerative colitis is not significant here, whereas in Reg 3A, the comparison between normal and ulcerative colitis is significant. So there is less expression of this gene in ulcerative colitis, which means that it could be involved in mucosal healing, but it could also contribute in the other direction, contributing to inflammation.
I'm sorry, I was mute again. Okay, so just for your information that you want to reproduce this graph, you can go to SmartBook and then there is a button for ANOVA analysis right here. For this specific experiment, we compare between different disease groups. So I select the disease one here with Crohn's disease, normal and ulcerative colitis, and then I run the analysis like that. Okay. Oh, I'm sorry. And then you can see the analysis result pop up under the ANOVA section. And this is one result that I had performed earlier. So besides the the cow box plot that we're showing you on the slide, you can actually see the like on SmartBook, the table showing all of the genes within this specific data set, and then you can have like a Manhattan plot for showing individual genes on different chromosomes. Okay, for example, let's say that we got the interleukin. Okay, let's say the interleukin 1. Okay, so here. Okay, just let's say the interleukin 1 alpha like that, and then you can see the gene box plot right here. Okay. Okay, now we go back to the slide and then we're moving on with another result. So we just perform ANOVA analysis, which is a very first round of analyzing this data set where we compare between multiple groups. But now we want to dive a little bit deeper to investigate the differentially expressed genes in Crohn's disease and ulcerative colitis separately when comparing to normal control. Okay. So in order to do that, we actually perform two separate DG analysis. one is between Crohn's disease and normal, and another one is between ulcerative colitis and then normal. Okay, and after performing the analysis, we can see quite similar result with the ANOVA, but in this case, we got the volcano plot for showing upregulated and downregulated genes in the disease. And when we look at the summary table based on the p-value and the log2 function, we have detected some of the most upregulated genes in Crohn's disease. As you can see, we got 4 genes here. And now let's say, let me go deeper into each of the genes here and what are those role in the disease. So first of all, we got the RQP9 or the aquaporin 9. So this is a water channel protein expressed in the GI tract. And based on here, you can see the upregulation of aquaporin 9 might be a response to inflammation or contribute to the alterated fluid dynamics in the gut. So basically there is some alter in interstitial permeability during Crohn's disease. And then to the second gene, the REG1B here, or the regenerating islet derived 1 beta. So this REG1B, as you may heard Julie earlier, this gene belongs to the REG protein family, which plays a role in mucosal regeneration and has an anti-apoptotic effects. So this upregulated REG1B might seem counterintuitive, as impaired mucosal healing is a characteristic of Crohn's disease. However, the increased expression could represent like an attempt to compensate for the ongoing damage, and or like a dysregulated response to inflammation. So yes, the upregulation of this gene in Crohn's disease is very noticeable. And after that, the other 2 genes you can see may be quite familiar to all of you guys. So the S100A8 and the CXCL5, they all related to inflammation state. So the S100A8 is like a calcium binding protein that involved in the inflammatory processes, and the upregulation of this gene is consistent with its known role in promoting inflammation and immune cell activation. So this likely contribute to the inflammatory cascade over like observed in Crohn's disease. And in terms of like CXCL5, this is a chemokine that attract neutrophils to the site of inflammation, and increased level of this gene likely uh, contribute to the recruitment of neutrophils to the gut mucosa, where they, release the inflammatory mediators and further exacerbate the inflammation. Okay, basically, like for a little summary, in Crohn's disease, we see increased inflammation, we see impaired mucosal healing, and also altered interstitial permeability. Okay, and now we move on to ulcerative colitis to see what do we have, what do we see in there.
Yeah, so again, we performed the same DEG analysis in ulcerative colitis, and we were able to extract out the top upregulated genes that were found to be more upregulated in our diseased group. So the first gene that we're looking at here is duo XA2. Oh, sorry, can you go back one slide, Hazel?
Oh, yeah, sure.
Yeah. So this
Mhm.
duo XA2, we see this upregulation, very high upregulation of this duo XA2 gene, but very significant downregulation in normal. And then to the next slide.
Okay, sure.
So, okay. Yeah, so here this duo XA2 gene is an enzyme that generates reactive oxygen species in the gut lumen. And our analysis revealed that this upregulation in duo XA2 in ulcerative colitis, and while a reactive oxygen species can contribute to inflammation, they may also play a role in host defense. So because we see this increased expression of duo XA2, this may reflect an attempt to kind of control bacterial populations or a dysregulated inflammatory response. The next gene, SL6A14, is a broad spectrum amino acid transporter. We observe significant upregulation of this. And this could be linked to altered nutrient uptake by inflamed cells, or it could be changes in the gut microbiome that influence the amino acid availability. The third gene, DEFB4A, encodes human beta deficin 2, which is an antimicrobial peptide. So the increased expression of this gene may likely reflect an enhanced immune response to changes in the gut microbiota, or could be a direct defense against invading pathogens in the inflamed colon. And then lastly, SERPIN B7 is a serine protease inhibitor. And the upregulation in this gene could be involved in various processes, including inflammation and tissue remodeling. So this could be contributing to a regulatory mechanism to control protease activity and limit tissue damage in the inflamed ulcerative colitis colon. So to summarize, some of the functions that we see related to these four genes are related to dysregulated inflammatory response, changes in the gut microbiome, increased inflammation, of course. Yeah, to summarize all those functions. Oh, you're muted again, Hazel.
Oh, yeah, I'm sorry. Okay, so before we move on, we got one question from Pranav that I would like to answer first. So the question is there any plant-based data sets? So on SmartBook right now, we believe that we not yet have the plant-based data set. However, our database is constantly, growing and growing. And then we will note it down as like maybe a request from users, and then we'll make a plan for if there are any other species, not only plant, but like beside human and mouse, we will have more species to come to SmartBook. Okay, so yeah, that's all. Okay, so let's move back to our presentation right here. Okay, so after we have identified the differentially expressed genes in both Crohn's disease and ulcerative colitis, we now want to understand the broader biological context of these changes. So to do this, we perform pathway analysis to identify commonly upregulated pathway in IBD. Okay, like this. So as you can see that our previous DG analysis identified distinct sets of differentially expressed gene in Crohn's disease and in ulcerative colitis compared to normal control. However, we were particularly interested in the genes that are commonly dysregulated in both conditions, like in the combination of the two diseases, as these might represent like core pathological mechanisms in IBD. So to visualize the overlap between these gene sets, we use the Venn diagram, like a Venn analysis, as you can see on the figure right here. So there, the intersection, right here, the orange part between the normal versus CD and normal versus UC, okay, contains about 5,000 are the genes that are like differentially expressed in both diseases compared to normal. And these genes are the genes that we focus on for our subsequent pathway analysis. And when we perform the pathway analysis, we perform on multiple databases at a time to compare between different databases and then also to add more layer of information to our results. Okay. So, okay, and then we can, this is the result of commonly upregulated pathway in the first database that we use. Okay, so Julie, can you please go on and explain people about the result here?
Yeah. So following differential gene expression analysis and then Venn diagram analysis, we can further perform pathway analysis to understand the functional implications of some of the most commonly dysregulated genes. So the slide right now is showing enriched pathways in the Go biological processes database. And the type of graph that we're looking at here is called a dot plot. So the dot plot displays the gene ratio, which is the overlap of genes from our list of DEGs versus the gene sets in the Go biological processes pathways. And then it's colored by the adjusted p-values for each of the enriched pathways. And as you can see here, the top enriched pathways are strongly related to immune function, including positive regulation of cytokine production, leukocyte-mediated immunity, adaptive immune response, and several pathways related to B cell and immunoglobulin activity. So this convergence on immune cell or immune-related pathways strongly supports the hypothesis that this regulation of the immune system is central and a shared mechanism in both Crohn's disease and ulcerative colitis. And so therefore, these findings may have implications for the development of therapeutic strategies that target these shared immune pathways.
Yes, so that's like the upregulated pathway in GoBB database. So basically, we all see like immune related pathway that like again strongly support the hypothesis that this regulation of the immune system is like a central and shared mechanisms in both of the two diseases. And these findings may have implication for the development like of therapeutic strategies that target these shared immune pathway or even target the previous commonly upregulated genes. And then when we move on, like we try on with another database, we also see the same result like we see in the DGN database. Most of the time we see like really high upregulation of inflammation, infection, and then we also see the upregulation of inflammatory dermatosis, inflammatory disorder. So basically, again, add more layers to our result. Okay, and then okay, and now we come to the last but not least part of the analysis, where after like a very streamlined experimental design, like from the ANOVA, from the DGE to the pathway enrichment, finally we come to the deconvolution, where we figure out like what cell types involved in the mechanisms of the two diseases. Okay, so in this part, Julie will explain to all of you guys like what cell types do we see and how different between normal versus diseases one. Okay, yeah.
Yeah, so the cell type deconvolution is the newest feature on SmartBulk. And the way it works is that you can choose one of the models to use as the model for the cell type deconvolution. So there's different tissue types available. So here I broke down the deconvolution, or I performed the deconvolution twice, one using the large intestine model and one using the small intestine model. So as we know from the beginning, ulcerative colitis is only ulcerative colitis because it occurs in the large intestine only, whereas Crohn's disease can occur throughout the entire GI tract. However, through this specific data set, they took samples from the small intestine. So on the left-hand side, we're looking at B cells, and we want to look at Crohn's disease versus normal in the small intestine. And we can see an elevated fraction of B cells in our Crohn's disease versus our normal. On the right-hand side, we're looking at T cells. We see a very, very high fraction of T cells in ulcerative colitis compared to normal. And this is all to say that we see an overall elevated fraction of immune cells in our IBD groups. And this is kind of what we expect because IBD is an inflammatory disease. So we expect these elevated fraction of immune cells to be present in our IBD groups.
Okay, and then the next one, yeah.
Okay. And then in the next slide, so this is another finding. We see a decreased expression across both the small and large intestine, across Crohn's disease and ulcerative colitis, a decreased fraction of epithelial cells compared to normal. And this might be because depending on where these samples were taken. So, for example, if the samples were taken from sites of inflammation, where we expect to see a lot more immune cells happening at play, then we start to see less and less healthy epithelial cells that might contribute to healing the gut and healing the intestine. So this is the patterns that we see in the cell types.
Yeah. Okay. Okay, so that's like the uh cell type deconvolution that uh, we saw in this combining data set. Basically, we see more B cell, more immune cells in the disease, and we see less epithelial cells in the disease. So like a reverse trend. And that's also like all of our biological findings today. Before we wraps up this meeting, I would like to give some of the summary for the meeting. So basically, today we use like a bulk analytic data. We use like a combined data set where we perform like a very straightforward experimental flow with, first of all, we compare the whole expression profiles of of three conditions of ulcerative colitis, of Crohn's disease, and normal together to figure out some of the commonly enriched genes in IBD. So we see the upregulation of ABCB1, of interleukin-related genes, and also of the REC protein family. And after that, we dive deeper into comparing Crohn's disease and ulcerative colitis with normal control individually. And then we found some of the top differentially expressed genes in this condition. As you can see in here, we got the aquaporin 9, we got the REC1 beta, REC1 alpha, and MMP3 for Crohn's disease. Then we got the DOXA2, the DEF-BA4A, the Serpin B7, and the ABCA12 for ulcerative colitis. Then we dive deeper into the commonly enriched pathway for both of the disease, where we found the enriched positive regulation of cytokine production, leukocyte-mediated immunity, adaptive immune response, and are enriched, and several other pathways related to B cell and immunoglobulin activity. And then the last thing, when we perform the cell type deconvolution, like align with our like pathway enrichment finding, we see more of the immune cell, the T cells and B cells, and due to the disease condition, we see less epithelial cells in ulcerative colitis and Crohn's disease. Okay, and that's all for our um for our club talk today. Before we move on to the Q&A section, we would like to have like a short, like very short survey for you all, because today we introduced you to the SmartBook, our book and analytic platform. So we would like to have some feedback from you guys, like what kind of biological data that you're interested, and then what analysis features that you want to be included in SmartBook, so that we can include it into our development cycle, and then we bring it to you as soon as possible. So please let us know, and then I will give you guys like around 1 minute to fill in the survey. Okay. Okay. Hmm. Okay, so that's for the survey. Now we move on to the Q&A, where we we have received, please feel free to let us know if you have any questions. Put it in the Q&A box right here and we will answer it right away. And okay, so for the first one, we got one question from Nishat Fatima. So Nishat asks us like the elevation of T cell should be commensurate with patches appear in the large intestines. Did the study carry out spatial analysis that consider where T cell proliferation occur in UC? Oh, this is a very interesting question where we got, we got more information from spatial context, but up to my knowledge, I don't think that in this study, the author like did perform any spatial analysis. If we want to perform like spatial analysis um for IBD, I think we might need more information, like more studies that, really focus on the actual cells on like a spatial on a tissues so that we can know whether like the patches appear in the large intestine or not. So yeah, that's my question for now for the spatial analysis. And then about the other question of the log 10 and log 2 normalized data, okay, I'm sorry, let me put answer line right here. We will follow up with you through email and then we will share with you like a very detailed information on like how we perform the normalization prior to the ANOVA and the DG analysis so that you could get the like a clearer view on why do you see log 10 and log 2 normalization on our plot. Okay, so thank you for your question. Okay. Okay, we got another question from Shilpa, Shilpa. Oh, I'm sorry if I pronounced your name wrong. Okay, so hi, I'm interested in to learn how to set up the analysis in Bioturing. Do you have any video links that can help me to learn how to perform such analysis? Oh yeah, sure, we have. So if you want to learn more about SmartBook in specific, our college will share with you the link to our video like YouTube channel and also to our guideline page so that you can learn more about it. And for Shipa and for all of you guys here, we are very happy to set up like one-on-one meeting with you guys so that we can show you like specifically how to set up analysis, how to perform analysis in SmartBook and also in every other platforms that we got in our ecosystem. Yeah.
Yeah, so we hope
Okay.
this analysis was useful to any of you who are performing any bulk RNA sequencing analysis, because this gives you a good overview of how you would approach your analysis, going from QC to going through the DEG analysis, through the Venn analysis, and then through the pathway enrichment, and then finally deconvolution.
Yeah, sure. Okay, so Shipa, I can see that my college already put the SmartBook document link to the chat box, so please visit there. And then yeah, and then if you want to book one-on-one meetings, just please let us know then. And yeah, oh yeah, so sure. We will send you the link to set up like one-on-one meeting. And yeah, we'll see you later. Okay. Okay, that's done here. Yep. Thank you so much. Okay, we done here. And now please let us know if you have any other questions that you would like to answer. Okay. I will wait for like maybe 2 minutes more before we wraps up this meeting. Yeah. Mhm. Okay. Okay, so if we got no more question, this is also the end of the club today. And see you next week. Yeah. Okay, so see you. Oh, for Nisha, yeah, we will follow up with email. So thank you a lot. Okay, bye.